human tissue is one of the most valuable substances per gram on planet Earth, like in the mid-tens of thousands of dollars per gram, if you could produce it. There's one system that has ever produced tissue, one engineering system, human development. And in the past, 3 years, the data sets that could provide a fuzzy representation of how that happens have come online, such that you can use that as basically a prior to ground models that will then design experiments to be done in the wet lab. We did this with a team of 5. And having 2 robust tissue engineering programs with a team of 5 if you went to a normal tissue engineering team, I think they would say you're off by a factor of 5 there. I think they would view that as slightly insane. We think there's a strong possibility that this is a winner takes most, if not winner takes all market. I think that each incremental tissue that we add is going to be easier and easier and easier. We could, at the limit, produce any tissue. If we're successful, the organ shortage shouldn't exist anymore.
Welcome to the Free Radicals podcast, where we interview the scientists and builders working to dramatically extend human lifespan and bring about a sci-fi future where humanity has full control over biology. Today's guest is Matthew Osman, co-founder and CEO of Polyphron. Matthew Osman is a twice-exited tech founder turned biotech founder. His company is building a computation-first tissue foundry that applies AI and lab automation to create human tissue at scale. If they can achieve their grand ambition, it could mean an end to long organ donor lists and therapies that could dramatically extend lifespan. Listen to this episode to learn more about the core bet and technological platform at Polyphron, the remarkable progress they've made on 2 programs in heart and brain tissue in just a year, and what's next for the company. I am your host, Daniel Schur, and my co-host is Eric Dye. I hope you enjoy this conversation. Welcome to the Free Radicals podcast. Today we have Matthew Osman, founder of Polyphron. Welcome.
Hi, good to be here.
Good to have you on. So we wanted to start out with some background on Polyphron. So in early December, your company came out of stealth as the first scalable tissue foundry. So can you tell us what that means and what is Polyphron?
Yeah. So Polyphron is an autonomous tissue foundry. So we're attempting to produce human tissue from iPSCs at scale and at incredibly high margin. If we had this capability, it would unlock a bunch of kind of downstream use cases, which we could chat about, I'm sure. But functionally, the company is built on the premise that human tissue is one of the most valuable substances per gram on planet Earth, in the mid-tens of thousands of dollars per gram, if you could produce it. And we think we have found a way to use closed-loop computation, automation to crack some of the issues that have plagued the tissue engineering field for 20, 30 years. So the aim is to produce human tissue eventually of any size and complexity and geometry. And there are therapeutic applications and also applications as a high-fidelity screening tool, because it would be obviously the most perfect model to figure out what's happening in human biology before you would go into kind of animals or even into human trials. So very compute heavy, very automation heavy. We picked the name Foundry quite specifically. We're obsessed with cost control, and we consider this to be closer to a semiconductor fab than a traditional wet lab discovery project. Of course, we have a wet lab, but we've kind of built the entire team and the organization around this idea of reproducibility and scale.
Awesome. So for folks who aren't familiar, right, it's very hard to grow tissue. It's very hard to grow organs. Can you talk a bit about what does that space look like currently, and what's the problem Polyphron is trying to solve there?
Yeah. So there are kind of a number of approaches that have been tried before in tissue engineering more broadly. You can kind of put them into a series of buckets. So one would be kind of organoid type approaches that have been, active and relatively well funded over the past decade or so. So that is where you're basically trying to take the cells that you would find in a specific tissue, have them self-assemble, and use that as a model to be able to study biology and maybe transplant. You have bioprinting techniques, which is where you try and kind of reproduce the geometry of tissue through, maybe extrusion-based or other-based bioprinting technologies. You have scaffolding-based techniques. You have xenotransplantation, is I suppose broadly in the attempt to try and produce tissue where the bioreactor is an animal. Most of these approaches rely on a mechanistic understanding of like how tissue development happens in practice, and they're enormously bespoke very often, pretty artisanal. It's hard to see how the unit economics works, and the fidelity is fairly low. So, just taking the organoid approach, the organoids, as a rule, tend to be very, very bad at reproducing or recapitulating structure of tissue. So you might get the right cell types in an organoid, but you're not going to get them in the right structure. And one of the premises of what we're doing as a company is that structure really, really matters. And it's the first thing that you should nail. So like native morphology, native structure, particularly in tissue types where function is downstream of that structure. So in excitable in excitatory or contractile tissues, so like cardiac tissue or like cortical tissue, for example, unless you can recapitulate the structure of the underlying tissue, what you're going to get as a unit is going to be a very, very poor model for testing. Structure is extremely important. And it's important for trying to simulate function and also trying to restore function. So if you're ever going to use these kind of tissue-like products to as a screening tool or as a therapeutic tool, you need to be able to recapitulate the structure of a tissue. So within like cardiac, for example, if you cannot reproduce the structure of native myocardium, so like myofibers aligned, what you'll get is like aberrant connectivity, and you'll get like arrhythmias if you try and use it therapeutically, and you can't test arrhythmias if you're using it as a screening tool. So in all of these cases, the first problem that we're trying to solve is, can you recapitulate the structure of tissue using this kind of closed loop system that we've built?
We're going to dive a lot more into the technology, I think, later in this conversation. Super interesting. We want to understand a lot better and how you think about it and the business model as well. And before we dive into that, could you just give like a bit more of a description of like human tissue, extremely valuable. Like, how would it be used therapeutically? Like, what's the world you're envisioning in terms of, partial tissue replacement, so people understand, what kind of therapies we're talking about?
Yeah. So when we look at indications that would be interesting for kind of partial tissue replacement, we're looking at initially indications where there is an existing reimbursement code that kind of feels similar. There's an existing surgical workflow. And most likely there's a transplant at the end of it as kind of the end point of care. So to give a concrete example, left ventricular tissue grafted after an acute MI, a heart attack, myocardial infarction, or as a kind of bridging deferral strategy to prevent someone needing an LVAD. So the idea is that if you could intervene much, much earlier with smaller chunks of tissue that are kind of functional units, you can prevent someone from having the kind of chronic organ collapse that would need them, that would require them to have an organ transplant. So the aim there really is to sort of solve the organ shortage problem, not by engineering organs, but by creating functional units of replacement tissue where you would intervene much, much earlier in the process. That has a number of advantages, of course. One is that you have a smaller amount of biomass to produce, which helps from a COGS perspective. The other is that you have a much, much larger patient population. So transplants are broadly ineligible for large numbers of people because of comorbidities and like a bunch of other reasons. we think that these grafts could be placed either as an adjunct to an existing surgery. So we're looking, again, to try and piggyback off an existing surgery, or kind of minimally invasively. And if you think about the alternative, so imagine that you could produce full organs. There's a magic technology that can produce full organs. Because of the way that those have to go in a human, that will not be the first line indication for someone who has, mid-stage heart failure, because you have to cut open someone's sternum and you have ICU time and you have a huge amount of downstream costs for the payer. So what we're looking at is smaller grafts of tissue that go in much, much earlier. So like a pancreatic graft in acute pancreatitis, kidney graft before you get to end stage kidney disease, partial liver resection, partial liver replacement, which you actually already see on the transplant side, and then cardiac, which is where we're spending like a lot of our time thinking about. It is a very, very good kind of entry point from an indication space perspective.
And so, you're not traditionally or originally from biotech. You're a twice exited tech founder. Can you tell us about what made you pivot into biotech and what that process has been like?
Well, the process has involved a lot of reading. So I spend a lot of my time reading papers and textbooks. So I've been kind of interested in the concept of functional tissue replacement, partial tissue replacement, creating tissue for a long time. What really prompted the change was a personal situation. So I had a medical issue that would have been easily resolved. It ended up being fully resolved, and I'm perfectly well. But it would have been easily resolved if we'd actually had like a gram of pancreatic tissue that that could have gone in. And so then I started thinking about what are the ways that we're attempting to produce tissue right now. I linked up with my co-founder Fabio, who's the CSO and also heads up all of our computation, who's a computational biologist and tissue engineer by background. And he'd been thinking about the same problem that I had, but from the technical side. And the big unlock that made us kind of comfortable starting the business is that ordinary ways that you would make tissue involve like extremely intensive labor intensive design of protocols and testing, and normally like mechanistic understanding of of like the pathways involved in getting to tissue. Our thesis was that there's one system that has ever produced tissue, one engineering system, human development, and in the past like 3 years, the data sets that could provide like a fuzzy representation of how that happens have come online, such that you can use that as basically a prior to ground models that will then design experiments to be done in the wet lab. So that was the hypothesis we went in with when we started the company. And we published a white paper a couple of weeks ago that kind of shows that that's borne out across multiple tissue types. So that was another thing, is that most teams are going kind of tissue by tissue. So ordinarily what you would, if you were going to create a tissue product or attempt to, there would be a PI in a Stanford lab. You would have a huge number of postdocs kind of all trying to understand different bits of the cascade of morphogenesis of that tissue. And the approach that we took was, well, actually, a pan-tissue approach makes more sense because actually morphogenesis happens across all tissues, in a sort of a connected way, and that there would likely be latent rules of morphogenesis that are illegible to a human, but legible to like some of the new model architectures. So we took the leap just under a year ago. And then, we came out of stealth in December, I think.
So just to dig a little bit deeper into what you're describing, effectively what you're saying is that there's a way to measure a lot of the latent features of tissue morphogenesis in this closed loop model system that you have set up for tissue engineering, and you're calling this a tissue foundry. Can you help us understand how this differs from the state of the art from other existing tissue engineering approaches?
Yeah. So I think a couple of things. One, the closed loop capability is critical to making this work. So we're running these experiments semi-autonomously right now, and at like very, very high throughput. So we produced close to 1,000 of these tissues in the past couple months. The system is built to require no priors about development in that tissue type. So normally you would have to be a developmental biologist with an expertise in the tissue type. We're trying to build a generalized system. We have people with expertise, obviously, in developmental biology, but they're not involved in any of the hypothesis generation or selection. That's all done basically with the model just using development as a prior. And the other thing is that the readouts that we have are deliberately non-destructive. So on one side, we have this kind of developmental atlas that we've created. That is basically the set of priors, and that is connected through a model and a series of models to 3D iPSC cultures that are then read out non-destructively. Right now, we're using Brightfield. We're adding other non-destructive readouts, and you essentially iterate your way closer and closer and closer to tissue. So we have, relative to sort of state-of-the-art approaches, I think we're the first to show that the platform can produce tissues from two different germ layers that are pretty different. So heart and cortex. We're the first to show that we can recapitulate near native morphology in a very, very short period of time. So in our cortical program, the native in vivo orientation of the neurites is about 90 degrees to the basal, maybe apical actually, surface. Our model in 3 iterations platform got us to 82 degrees. So it's pretty close within the margin of error, honestly. And then we replicated that in the cortex in cardiac for a cardiac program. And one of the key things that we have done compared to sort of a traditional set of approaches where you would have these kind of bespoke assays and this very artisanal setup is each loop is getting quicker and quicker. So the cortical program took 10 weeks from start to finish. The cardiac program, we basically transferred the model with minimal retuning, and that took 5 weeks from start to finish. And if we transferred that to another tissue type, we anticipate that would get even quicker. And so the fact that it's pan tissue, the fact that as far as we're aware, it is certainly, like, if not the first, it's definitely the state of the art level of control over tissue morphology, and the fact that we're doing this without needing mechanistic understanding of what's happening in morphogenesis other than what you're giving a model. All of that exceeds state of the art in terms of like the platform build out. And then in terms of results, unlike other approaches, we're actually recapitulating the structure of natural tissue. And that's a huge deal from a screening perspective and from a therapeutics perspective. So all of that in the whole, I think, represents a significant step change in what's been done before, and I think a really interesting approach to the problem, and we'll see how far it scales. But right now it seems to scale pretty nicely. The next program, which we haven't announced yet, we believe is in the tissue type that we're working with, the most complex structure that anyone has produced from an iPSC. So we're able to scale up complexity and not just across tissue type. And so that's super exciting. And we'll publish a white paper. We're trying to build in public as much as possible. So we'll publish a white paper about that in the next few weeks.
And you got a sneak peek on the Free Radicals podcast. Scoop
Yeah,
right here.
Yeah.
One
That's
question. One quick question about the way the model is helping to improve the experiments. Like, what are the parameters the model is informing? I know there's one set of like, what are the ligands that you're sort of putting into into the dish where you're growing these? Like, what are other things that it's telling you how to intervene?
Yeah. So right now we are intervening. The kind of the parameter space broadly is the composition of the ECM, so the extracellular matrix. So right now you can think of these as kind of cubes with iPSCs in that we do stuff to, and those perturbations right now are primarily small molecules, peptides. Parameters, of course, like concentration of those, timing of those, the search space, even with like triplets of small molecules, explodes pretty quickly. And so what is great so we have this active learning approach that right now is using Bayesian optimization. It's able to compress the search space really, really fast. So we're getting like a 99% compression of the different combinations across 3 iterations. As we build this out, we do notice sensitivities between tissue types about what kinds of perturbations get them closer to native tissue. So we will add on additional read-write functionality that will allow us to do non-soluble interventions. So like MEA, like electrical stimulation is one. Right now we play with, we have a parameter around, shear stress and using kind of fluid dynamics to do that, but there might be another way of imposing kind of physical constraints on the system. And then at the limit, we would probably consider having, more hardcore interventions than what we have right now. But you'd be surprised at how far you can get with combinations of small molecules and peptides. Not all the way, to be clear, but the trend is like extremely encouraging. And of course, you have to remember that the only engineering system that has ever produced tissue is doing about the same thing. It's a bounded space morphogenesis, which is good because otherwise it might be an even harder technical problem than it currently is.
One question that comes to mind for me is this real challenge of working in general in these very high degrees of freedom search spaces, right? Which is you have a lot of parameters that you can tune to modulate the search space. And then also there's a lot of different metrics that you can measure. And especially with human biology, there's this challenge where you're not even sure if what you're measuring is a true reflection of the principal components of whatever system you're trying to recreate, right? And so that's like, how do you think about the observability challenge of what kinds of data matter, and am I collecting enough of the right kind of data? And then two, well, let's just start with that question. Like, how do you think about the observability challenge?
So it's obviously something that, from a technical perspective, we consider consistently, particularly when we think about multimodal readouts. One of the reasons that we started with structure, beyond the fact that if you can prove that you've already beaten organoids, which we believe we have, is that bright field morphology structure is pretty observable, and you can compare it to in vivo like very, very easily. Obviously, as you go up in complexity, that might not be sufficient. So like metabolomics is something that you can do kind of non-destructively by analyzing spent media. There are a number of other approaches that can kind of tell you roughly close you're getting to your target. We are in the process of designing some custom hardware to include like the additional data modalities that we think are going to be required to have like the sufficient level of observability to solve these problems.
And maybe going to the other side of the coin, how do you think about which parameters are worthwhile to search along, right? So you have like 24 different molecules that you're starting in your search space, right? But how did you select those 24 molecules? And how do you think about not arriving at some local maxima of tissue recreation?
Yeah. So I mean, that's the the beauty of using the kind of the class of models is the explore versus exploit dynamic. And so that's kind of built in. And we kind of use that dynamic to try and make sure that we're not in local minima. And then obviously we have like ground truth, which is in vivo. And yeah, so broadly, that's kind of where some of the secret sauce is in. It's actually, part of the core like ML challenges that we are trying to solve at all points. We are hiring good ML people. If you want to come and build tissue, come work with us. It's my shameless plug.
One sort of, when I think about this problem, like there's something interesting here, which is if you think about what's happening in an embryo, like there's a lot happening, right? Like there's so many different signals that are being transmitted that are leading the cells to grow in a particular way. But like, when you're trying to recapitulate this in the lab, you're trying to compress all of those signals down to, I don't know, like the 20 ligands that matter. Do you have, I don't know, like what got you to believe that there's a way to sort of compress the parameters down to something that is manageable, like by an AI in the lab?
So one, I suppose, is that like morphogenesis is the existence proof that like it is possible with a constrained number that isn't 100. There's a really beautiful Alan Turing paper about this from I think the 1920s or something. That was the core technical, the first technical risk of the company was that that's even tractable. It might have been that it was impossible to compress the search space. It might have been that we got stuck in local minima. It might have been that playing with these kind of 20 or 24 in the case of the cortical program wasn't sufficient. But so far the data sort of suggests it was. So the candid answer to the question is that was like hypothesis one that we needed to de-risk, and there was somewhat of a leap of faith, we turns out to be at least directionally correct so far.
When you think through what the core technical pieces are at Polyphron, how much of the tech stack at the company was necessary to build from 0 to 1, and how much of it was stringing together components from existing tools, hardware, software, services? And how did you think about the build versus buy mentality as you constructed the company?
Yeah, so something just to kind of point out, because I think it is important. We've done this in an extremely capital efficient way, and with a very small team. Like we did this with a team of 5, and having 2 like robust tissue engineering programs with a team of 5 is, if you went to a normal tissue engineering team, I think they would say you're off by like a factor of 5 there. Like I think they would view that as slightly insane. So the way we think about things is kind of eventually this whole thing is going to be kind of fully autonomous. Ideally lights out. Initially, what you have to build is point automation where it matters, and a loop that is kind of, at least in principle, automatable. Because we're trying to do this very, very fast and in a capital efficient way, we try and use either retrofitted or existing components on the hardware side for now. So we're using a commercially available microfluidic chip at present. As we add on a different modalities that we think are going to be essential for us getting to our desired phenotype, we will have to custom build, 3D print or something, our chip to be able to handle those modalities and have those readouts. On the technical side, you can. There's a sort of an important distinction to be made about the data sets that we're using as the prior, which is that the primer data set is this developmental atlas. and we're standing on the shoulder of giants there. There are consortia who have spent years and a lot of public money very often building these, and we're extremely appreciative. That most of our data, most of the kind of the pertinent data, the load-bearing data, is the data generated from the lab. So you essentially this is not quite correct, but like naively, you can think about the developmental atlas is kind of like constraining the manifold in some way, and then most of the data is coming out of what you see in the lab. So we initially have started with trying to use as much stuff that already exists as possible, but I think as we get larger, as we build this out even more and we get kind of more and more complex, we're going to have to build more custom instrumentation or kind of retrofitted instrumentation. Yeah, I'm a big fan of vertical integration, just generally.
That's awesome. That was super impressive. Like a staff of 5 to get to this level of progress in around a year is like, it's incredible, right? So amazing progress.
Thank
you. Curious as well on how you arrived at this question of starting autologous versus allogeneic.
Yeah, I mean that is the question I think you should ask anyone who's working in something cellular, single, all the way through to multi and structure, which is what we have. So I think so from a technical perspective, we are basically agnostic. So the platform itself is designed to be able to reach the same endpoint regardless of what your starting cell line is. And there's some data that we're going to publish on that as well. So that's another kind of important thing about the difference between us and existing systems of trying to make this work. So, existing tissue engineering approaches is that once you have a protocol in a standard tissue engineering approach, it's extremely brittle. And trying to transfer it to a different starting iPSC line or donor normally breaks it. And we have built a platform that is designed to be invariant to starting donor heterogeneity. So in practice, allogeneic versus autologous becomes a COGS question and a availability, like a logistics question, versus a technical one. So we're in the fortunate position that for certain indications where autologous would be extremely valuable to the end patient because of not needing chronic immunosuppression, we have a partnership with Celino, who has this amazing technology. You should talk to Nabia if you haven't already. Amazing technology that automates the iPSC creation process. So we don't touch reprogramming. iPSCs are the feedstock into the Foundry that get then turned into the chunks of tissue that you care about. So we have an amazing partner on the manufacturing side, and they're starting to deploy in hospitals, I believe, soon. And so you could imagine, point of care or autologous. But likewise, on the allogeneic side, you could do a couple of things if you have this invariant system. One is you do everything autologous, which if you do everything autologous probably causes some costs and logistic issues. It's probably very valuable for a specific subset of patients. On the allogeneic side, you can hypothetically produce like multiple SKUs of the product connected to an HLA subtype, and the number of SKUs that you would need to make to get 70-80% coverage in the US is a lot, but not insane. So it's like 1 to 300, something like that. There are, however, markets where the heterogeneity is significantly less, where you can get coverage of most of the population with like fewer HLA subtype lines. So Japan is one where I think you need something like 10 to 20, and you can cover 70-80% of the Japanese population. And they also have a huge history of regenerative medicine. So the short answer is it's going to be extremely indication dependent, and it'll be fundamentally a kind of a cost decision, like a trade-off. But the platform itself is supposed to be kind of designed to be invariant in a way that would allow us to do it either way. So the TLDR is we're pro having cake and also eating it when it comes to allogeneic versus autologous.
It'd be great to go into some of your initial compute-first paradigm experiments, which you recently published, I think, a couple of weeks ago. But help us walk through some of the design heuristics here and some of the results from these first experiments on excitatory neuron and cardiomyocyte alignment.
Yeah. So the two programs that we kind of shared initial results on, just to kind of bring everyone up to speed, as you point out, one is in the cortex and one is in left ventricle broadly, myocardium. I mean, it's in iPSCs that are potentially progenitors to that. It's not in the final tissue. And we focused on cell alignment and polarity as our optimization target because, as I mentioned, we believe it's a precondition for like higher order structure, and showing that you can kind of control that already gets you above and beyond existing approaches that involve clumps of cells just kind of lumped together. And so in cortical tissue, you have a neurite orientation that is broadly like 90 degrees, and in cardiac tissue, you need similar style alignment of myofibers in order to produce kind of contractile force and also get kind of the correct conductivity. At a sort of simple level, the one way to think about it is because these are like electrical circuits, you need to have like correct alignment because otherwise you mess up the circuit. Now there was another. So we essentially had the closed loop platform focus on two objectives simultaneously. One was alignment magnitude. So how aligned are they relative to like what we see in vivo? And the other is how consistent was that? So how consistent is the alignment across the construct? Because one of the big issues with organoids and like organ on a chip systems and like a bunch of other systems that have attempted to recapitulate human tissue is drift and reproducibility and variability, and like you need to be able to show control over that, both for the therapeutic use case and also for like a screening use case. And so in the cortical program, we were attempting to get the platform to reproduce autonomously the in vivo like radial orientation that you get of a specific neuronal subtype. So that was something else that we wanted to show, is that, it would be if the platform was making every single cell in the culture aligned, that would also be bad. It is only a specific kind of neuron, an excitatory neuron, that has this alignment feature. So we wanted to show in the cortex, can we not only get the in vivo alignment of these cells, but can it be just for these cells? Because there are probably ways that you could kind of get every cell to align that would also cause issues. And so we ran 3 Bayesian optimization iterations. The kind of base case control, which is roughly what you would get in an organoid, is like 45 degrees on average. So across the construct or across kind of a section of tissue, you would see some neurites pointing like this, some pointing like this. It's basically 45 degrees on average. And across 3 iterations, we got it to, I think it was 81.9. And in vivo is about 90, plus or minus 5%. So we whittled down 20,000 genes to do that into 24 candidate morphogenic interventions. Those were applied partially using a liquid handler and then partially using some humans in the loop. And then we read out with bright field, and over the 3 iterations, we kind of, you could essentially see the neurites like pointing up, which is what you would expect, and just those neurons. So that demonstrated cell type specificity, which you need to be able to demonstrate as a platform, consistency, and then also in vivo alignment. And so then the big test was, well, the skeptic would go, okay, but maybe there's something specific about neurons that like makes them amenable to this approach. And so we said, okay, well, can we show the same structural control in another tissue type? And we picked cardiac for a bunch of reasons, including downstream applications and indications, but it's also a different germ layer. So if you're using kind of developmental references, you want to show that it's not something specific to the germ layer that you started with that kind of makes this a tractable problem. And so it was a distinct tissue type. We broadly applied the same model with minimal retuning. And then we got the median directionality in the top performing chip in cardiac, again, across 3 iterations, was 89.4. So essentially what we showed is this kind of like effective lab in the loop optimization where across 2 distinct tissue types, you can control one of the most important aspects of like structure that leads to higher order tissue organization. You can do it in a very fine-grained way, so you can specify specific cell types that you want to affect. And the iterations from the programs got 70% cheaper, I think, from iteration 1 to iteration 2, and also got faster as well. So as I mentioned, I think it's like a roughly 50% drop in how long it took. So we're very, very excited by this. Obviously, I think it's the most compelling example that we've seen of someone being able to make this problem look tractable now. And then we have existing programs that are will unwrap for the world soon around kind of laddering up in complexity. So can you replicate tissue, not just morphology, but like other aspects of tissue complexity that you see it in vivo? And we've also got some pretty exciting results there, as I hinted at before.
I'm curious what some of these results tell us about the nature of morphogenesis. So, if you can train one model that can work across many different tissue types, to me, it seems like you're recapitulating, I guess, like the language of morphogenesis, right? Like you're figuring out the shared language that all the different tissue types speak. And, is that how you would think about it? And also, how are you thinking about, I don't know, as you build the model, like interpretability? Like, are you hoping eventually humans will understand the language of this model too, to understand the biology?
So the second question first. One, from a scientific curiosity perspective, it would be great if this were fully legible. And I think you could build legibility modules in the same way that you like interrogate token prediction in an LLM. That's not a primary focus of ours right now. We care about whether you can actually create the tissue that that you care about. And again, I think that feedback beats understanding. There's a great one of the co-founders of Lila Sciences published a great blog post about this, and I think there is a danger in demanding mechanistic understanding of what is happening when essentially iterative experimental loops with wet lab validation will like get you the product that you want. And then the sort of the first question, our hypothesis is that there are latent rules of like morphogenesis that a model can learn, and it's why, from a company building perspective, we think there's a strong possibility that this is a winner takes most, if not winner takes all market. I think that each incremental tissue that we add is going to be easier and easier and easier. And we could, at the limit, produce any tissue.
So I'd love to talk a bit about that hypothesis around the business model. In your letter from the founders, you framed the business model in a really interesting way, right? By especially you made reference to some recent sort of discussions within the biotech community around, hey, why aren't there any trillion-dollar biotechs? Now we have Eli Lilly, but you talk a lot about the ways in which often pharma companies don't have a moat beyond the intellectual property, but you're describing Polyphron as having a moat within the model weights and the process. I'm curious to hear you elaborate on that more, and with the specific, I guess, question I have, which is like, where in the process do you see the moat developing? Right, like you guys have a team of 5, and you've gotten exciting results. You know, if the technology is ready, you could imagine a lot of different companies training similar models.
Yeah. So a couple of things there. So one is that as I mentioned before, but I think it's worth restating, the publicly available data is the primer. And so, hypothetically, that's accessible. You actually need to do the experiments. So you need a very tight wet lab validation loop. Most of the data that is pertinent for model training is happening there. That is not released anywhere. And we're not doing that. So you would have to, if you were kind of trying to come from behind, run the experiments yourself in the correct setup. So it's not sufficient to just like train a model on existing data. You would need to mainly do all the validation experimentally and use that to train your model. And then on the sort of the company building side, yeah, I mean, I think it's similar to I think process power in the sort of the 7 powers framework is stronger than patents pretty much all the time. So the way, I mean, anyone listening to this podcast will be kind of familiar with this, but the way that normally value is captured for a classic pharma intervention, so a small molecule, biologic, whatever it may be, is through, a state-sanctioned monopoly, so a patent. You file before approval. So even though the patent term is 20 years, in practice, you get about 12 years of sort of monopoly profits. And then they don't quite go to zero, but they go asymptotically to zero once generics, because generics are basically waiting for that day. And like the patent cliff is like a real problem. And it's a really, really hard thing to build a company that compounds where your winners reset after 12 years or so, and so the framework that we're thinking about this is that most of the hard stuff to copy is going to be in the processes around wet lab like in the automation, the connection between the dry lab and the wet lab, the sort of the weights broadly of all of these models. And those wouldn't be made public. And in fact, the IP strategy is likely going to be, prioritizing trade secrets over anything else. It's a different way of building a business, of course, but one I think that has a really strong shot of having a sort of a functionally different risk profile than you would normally have as an early stage discovery company, and maybe even different financing options later on, because your products stop having a should no longer be treated as like a DCF'd 10 to 12 years of cash flow, but almost as quasi perpetual royalty streams until someone puts a better product out on the market. And I think that opens up some like fairly interesting opportunities about how you would finance this company, how it should be valued, and like how value will compound.
And the model has an interesting component, right, where you mentioned it can sort of scale like a software company, right? In that, as you build this model, right, and you sort of invest the cost required to get it, you can maybe produce tissue at much lower marginal cost. And I guess I'm curious though, do you see Do you see this as a very high capex business? Like, is the big investment here getting the right talent to design the right experiments and get the right results? Or is it going to be kind of like, I don't know, a foundational AI company where, we're going to need like billions of dollars to like train models, right? To like run tons of experiments. Like, is there a path you see this going down?
We will be training kind of large models. There are cost components to this that are unavoidable. Like, you can do some stuff around like regulatory arbitrage and stuff. But there are going to be sort of hard limits there. I think that every single tissue construct that we produce makes our model better by some delta. And over time that will become like very, very hard to replicate. I think our manufacturing costs for these constructs will come down and down and down and down and down. And because of the way it's built, it is much, much closer to a foundation model company than a kind of a classic wet lab biology company. We are getting a lot of signal out of fewer tokens. Just to kind of, again, I'm kind of mixing a bunch of metaphors here. I'm using this as purely metaphorical. So I think it's very unlikely that our compute needs are going to be the same as training like a large LLM. But on the hardware side, obviously, automation is gonna cost some money. But I would mentally classify it more in the foundation model bucket than in the kind of like classic, wet lab preclinical biology company.
So one of the things we really like to do with this podcast is like we're bringing on people who are building the future, right? And I think it's very inspiring. So I would love to hear from you, like, what does the future world look like in a world where Polyphron is successful?
So I think if we're successful, the organ shortage shouldn't exist anymore. I think people, when they would otherwise have needed a transplant of a specific organ, will instead get minimally invasive interventions of immune-compatible tissue that help them stay healthier longer, and avoid hospital time, have more time with loved ones, and that is something that we strive for and we take very seriously at the company. But our aim here really is to attempt to kind of stop in its track most of the major mortality risks faced by humans, which at one point or another all tends to tissue breakdown. And that is the kind of level of abstraction that we work at. And so our hope is that we can head off that collapse with these partial minimally invasive replacements, and extend human lifespan, health span, whatever you want to call it, and that people can stay alive healthier and longer.
Yeah, we're at the end of our conversation. We didn't really use the L word, longevity. But I think it's clear that Polyphron is a longevity company. And yeah, it's a future, I think, where, I don't know, Polyphron is working on a really important pathway that can solve almost all of the issues of aging in theory.
Yeah, well, that's our hope.
Amazing. Thank you for joining us on the podcast.
Fun.
Cool.
This is great.
Thanks, guys.
Thank you for listening to this episode of the Free Radicals podcast. If you enjoyed this episode and would like to support us, the most helpful thing you can do is to share this with a friend that you think might enjoy it too. And please also leave us a 5-star review on Spotify, Apple Podcasts, and like and subscribe on YouTube. It would really mean a lot. I'm Daniel Schur, and my co-host is Eric Dye. Thanks for listening.